Okay, so this final part of this week's lecture is trying to get to the visual system. The question here is that how it's possible that the information in the brain is consciously experienced and um, accessible for further reports in some cases, like the Haynes visible case, but it's also not the case, like in the case of the blind side or a priming effect of the standard study. So to address this question, what we need to do is to look into the architecture of the visual system and then to consider how that relates to conscious experience. And this overall moves towards the study design or framework of neural correlates of consciousness. And that's going to be the main topic for this week and next week. So let's get started with the eye. So eye, as you might already know, that the lights are coming from this direction, from up, and then your iris restricts the opening of this, eye part lens, and then all of these lights goes into a straight line and then focused onto one particular part of the retina outside here. And then this is a lens and so on. Okay. So for our purpose, the most important component within the eye is the retina. The retina starts the visual processing by catching the light photons through photoreceptors. And the photoreceptor consists of cones that are responsible for color vision, as we talk about today, and then rods that are responsible for the night vision. It responds to the low intensity of the light. And that's what you can see in this area. Okay? And this elongation is the part of the rods and the cones. And then there are some layers of the intermediate processing, such as bipolar cells and amacrine cell. And in the end, it generates, it leads to this retinal ganglion cell layer around here. And then that is the first neuron that you encounter within the visual system that generate the spikes. Up until here, all of the computation within the retina is analog. And these retinal ganglion cells make a spike all or none, and then transmits information to the next layer of the visual processing. And the important thing to note is that the figure that I showed is, in a sense, inverted, upside down. So it's kind of confusing, but the light coming from up, and then the first thing you encounter is this axon bundles that is visible in this yellow area here, coming out from these retinal ganglion cells. So these guys actually send an axons, and that's the one that first sees, in a sense, the light. And then there are lots of processing layers here, and then the cones and the rods are located most close to the body, in a sense. And so light photons are dispersed and also interfered within this internal structure. It's not optimal to do the visual processing. The reason why this is evolutionary mystery, and it doesn't have to be like this at all, actually. And interestingly, octopus indeed has an, in a sense, optimum kind of visual processing architecture. So in their case, light comes here, and then first cones and the rod-like thing accumulates the light information here, and then the retinal ganglion cell-like structure processes the information and then sends an output into this way. So nothing interferes from the light, lens, and then photoreceptors. On the other hand, the vertebrates, the ones that have spines, in the back, they have all this architecture of initially ganglion cell layer that spikes, and then that sends an axon out from there, and then that is followed by the intermediate, structure, and then photoreceptors are located at the, back, and the information or spiking or neural transmission computation goes in this way, and so many of the stuff here potentially interferes with the light source. Okay. All right, and as a result of this architecture, the vertebrate species all have this phobia structure at the center of the vision usually. In the case of birds, they have different locations, but we'll not talk about it. The highest spatial resolution of the vision is attained in this phobia region. And the reason that you see some kind of dip here in this schematic is that at the phobia, all these axons are avoided, so that all these intermediate step of that could interfere with the cones and rods are minimized. And as a result, we have a better spatial resolution at the phobia or center of the fixation here. The definition of phobia, according to the Christoph Koch's book in chapter 3, is that the central part of the phobia, about 1 degree of visual angle in size, the width of your thumb at arm's length, like this. And it's around 1.5 to 2 degree of visual angle. And it's specialized to ensure that the vision there is as good as possible. So the useful number to remember is this concept of the thumbnail at the arm's length is roughly 1.5 degree visual angle. And now this study by Robert O'Shea shows that he asked many students to do this kind of measurement and then found that the variance across the people is very, very small. Thumbnail width, so everybody, pretty much male or female, if you extend the arms like this, okay, if you extend up like this, and then that width of the thumb, that corresponds to 1 degree of visual field. And that's where you can see and we can read, we can perceive everything best in a sense. Okay, so the distribution of the cones and the rods across eccentricities are not uniform. Here, eccentricity is defined as the angle from phobia. So phobia is always 0. And then here, for example, the light coming in here, and that's roughly like 15 degree or so. And then here, 14 degree is looking at something like this. So, as I said, the thumbnail at the, arm's length, that corresponds to, the projection of 1.5 degree, and that corresponds to this dip around the phobia. And then here, 0 to 1.5 degree here, you see that the density of the cones are very, very high per square meter. Okay, so given a particular area, number of the cones that you find is potentially up to 150,000 cones around the phobia at the direct fixation here, and then it degrades. Its density decreases quite a bit as you go to the periphery, but it doesn't reach to 0. Okay, that's also important. It's always above 0. But after 1.5 degree of phobia direction, region, it severely decreases. On the other hand, the rod increases. So the night vision is compensated by the rods, although we don't see much color from the rod. Another important thing that we will not go into the detail for this week, but next week we will go into this is this blind spot. So roughly 20 degree to 15 degree or so. Here, the other side, nasal side of the eye has this axon bundles that goes out from eye to the LGN. So this gray part is the one that indicates the axon, and these occupy quite a bit of this region, and around here we don't have any photoreceptors, and that's why you see this drop of the density into 0. So that's called a blind spot in the retina. And a blind spot, we don't usually see it because the other side of the eye see the information over there, and that is compensating. So the natural conjecture is that if you close one eye, then you see that blind spot, but phenomenologically you will not see it directly. And why is that? That's a question that we will discuss next week. But anyway, coming back to the another important phenomenological observation is next following. So just let's get this phenomenological inspection. Okay, just try to extend your arm from the display and then adjust the size of the distance from yourself to display so that this thumbnail image aligns to yours. And here, roughly, for me, this is a kind of a distance that aligns this thumbnail. Okay. Okay, now you're ready. Okay, so what I want you to do is to introspect what you see in the periphery. So just keeping your thumb at this thumb figure, and then closely look at all the without moving your eyes from the thumbnail. Can you see all the colored dots really clearly, or can you actually locate the location of the dots in a nice way? Can you count the number of the rows of the dots and the columns of the dots, and then can you see the red and the green very clearly in the far periphery? Okay, that's a first question. Okay, without answering to this question from my side, then I want you to think or introspect. What did you see in terms of color outside of the fovea? As I said, the fovea is around your thumb. Okay, and the cone density that is responsible for the color decreases quite a bit. And then if you saw anything clearly, sharply, richly, that's kind of interesting because the color vision supposedly is supported by the cone is decreasing quite a bit. And how rich, how sharp, is it actually corresponding to the expected level of the degree of the decay of the cone density? That's the question. And some of the philosophers, such as Dan Dennett, claims that based on this retinal density of the cones decline, everything in the periphery, if you think that the color is there, it's just illusory. And that's a philosopher's statement. Okay, and we, as a scientist, we want to test whether this is empirically true or not. But it turns out that it's not super straightforward to test it. So one more time. Now I want you to do it again. Align the finger here to the display, and then look at this figure. Okay. Can you now see the color of the red and the purple and the green and the blue in the periphery without moving your eyes? And also in closer to the fovea. So now this time, what do you feel? Okay, and then I want you to, if you have time, show this demo to your friends or family, and then ask their also color experience. And if you believe this cone density decrease corresponds to the color illusion of the periphery, then other people should also not see that color. But if there is something else, then other people would also see that the color is there. But this type of the introspection is just a first step, and we will discuss more into the detail to how we can confirm this type of phenomenological report or not by more sophisticated task. One possible way is the currently conducted experiment by Ariel in my lab. And if you follow this link, you will be led to this Inquisit website, and then if you press start, then the experiment will start. And it takes about 20 minutes to complete the experiment, and we will be discussing the results in the later week. So I encourage you to do this. Okay. So in summary of the last part of the lecture, retina in the eye is a sheet-like structure with cones and rods distributed unevenly across eccentricities. And I also asked you to introspect your color experience by having this fingernail thumbnail procedure. And we will discuss the implication of phenomenology further in the class and tutorials. And the next couple of weeks will be trying to address not only how the blind side works through the visual pathway discussion, but also what kind of phenomenological report we gain about color experience and why we can do that based on more detailed physiological investigation of the visual brain. Okay, until then, see you.